We're also not abandoning our drug screen. So currently, I think we screened over 4,400 compounds. We're going to continue to screen drugs. Obviously, the dream for everyone is to find something that's as safe as aspirin, but incredibly effective. And so we're continuing to screen compounds, and we will continue to do so until we run out of funds for those drug screening programs. All right, we're back in business. Now, to go back into the point of needing to identify additional topoisomerase inhibitors, our initial screen found one compound, and then we simply tried other topoisomerase inhibitors, and that led to about 16 topoisomerase inhibitors, all of which can produce the unsilencing. Next slide. There are also many other topoisomerase inhibitors that are being developed largely for cancer. So through a collaboration with Mark Cushman at Purdue University and Yves Palmier at the National Institutes of Health, these are probably two of the world leaders, they are two of the world leaders, if not the world leaders, on developing topoisomerase inhibitors for clinical use in cancers. Well, they're of course also thrilled that they might be able to be used for other indications such as Angelman syndrome. So they've given us access to a large library of topoisomerase inhibitors, and what you were seeing was an initial pass at testing 16 of these compounds or so for their ability to unsilence UB3A. So once again, anything above this white line shows that the drug is effective. As a benchmark, this is topotecan, which was our best compound before. So this is our best compound at its best concentration, and these are just a single concentration of these other compounds. And you can already see that there are 5 or 6 of these other compounds that are even better than topotecan, and we haven't even optimized them yet for dose. So now I think we're upwards of 30 different topoisomerase inhibitors that are effective at unsilencing paternal UB3A. So this gives us a large number of compounds that we can test to see which ones get into the brain the best, which ones are the safest, and which ones are the most efficient, basically. So we're looking for safety and efficacy and brain penetration, right? So these are all the types of things we can do now. Okay, next slide. Now, we really think topoisomerase is a good inroads to study for a therapy, because topoisomerases are expressed in almost every neuron just as UB3A is. And this is just a quick slide showing that the expression of topoisomerase is widespread just as UB3A is. Next slide. Now, the image, this is more maybe perhaps propaganda more than anything. This is an image to show you what we would hope to achieve. This is showing 2 neurons in the brain where you have a mutated or deleted UB3A one gene copy, and the other is a paternal silenced UB3A gene copy. And then what we hope to do is to treat with some type of compound that's going to kind of come in here. If this, let's see, let's give it one more try. With compounds that are going to come in like snowflakes, they're going to activate the paternal silenced copy of UB3A. They're going to turn it on, and they're going to make the neuron pink and happy, right? So that's really kind of the dream of what we would like to happen, to have a topoisomerase inhibitor or any other type of compound that could unsilence UB3A without causing undue side effects or adverse health events. So next slide. Now, it's come to my attention that there are a lot of parental questions out there about all of this. And I think often parents and caregivers might be afraid to ask me, and they shouldn't be afraid to ask me. And I want to take this opportunity to address some of the anticipated questions. And many of these were questions that a parent was kind enough to take off all the Facebook pages and synthesize for me in a de-identified way so that you can anonymously ask the questions that you've always wanted to ask. So I'm going to try to answer them now. And if I fail to answer your questions that I've anticipated, then pepper me with more questions. So next slide. So question number one, why did you only screen a few thousand compounds? Okay, why just a few thousand? We know there are drug libraries out there that have millions of compounds now. Why did you only screen a few thousand? Well, first of all, I will tell you that we really have the expertise of one of the world's leaders, Brian Roth, who runs a psychoactive drug screening program at the NIMH, behind us. He has access to huge drug libraries, okay, hundreds of thousands of compounds. It's not that we don't have the libraries to screen, it's the difficulty in doing so. It's incredibly difficult to screen neurons because these are non-dividing cells, okay? So we've actually performed one of the only successful neuronal screens to date. So if you search the literature, you will hardly see any drug screens done in neurons. The drug companies just don't do this because it's expensive and it's hard and it's difficult, so they've shunned it until recently. So we kind of estimate that if we were studying a dividing cell type, we probably, for the same amount of time and effort, could have screened millions of compounds. Okay? It's just that much harder to screen neurons because they're non-dividing cells than if we were screening dividing cells. If we were screening dividing cells, we take the cells, we divide them out infinitely, and we'd screen lots of compounds. Okay? The robotics can screen quickly. That's not the problem. It's getting the neurons. Okay? Next slide. So we focused on studying FDA-approved compounds, neuroactive compounds, ones that work in the brain, and known modifiers of chromosomes.
I'm hesitant because I know that somebody put a lot of effort into going through a list of questions, and I'm thinking maybe because we're tight on time, we put up each question, and then if it resonates with the audience and it's something that they want to know about, we stop and we pause on that one rather than going through, I don't know how many you got from the person who collated them.
I think I've got about 4.
4 is okay then. I was afraid it could be 20. Okay, I think we've got time for 4.
Yeah.
Can I just ask a question about whether UB3A, turning it on and off, could be tested in replicating cells instead of neurons as a way to
It's only silenced in neurons. So there's no way to turn something that's on that's already on in non-neuronal cells.
Okay, so I didn't realize that.
Yeah. All right. So we've tried to focus on compounds that if we found something that could move rapidly forward. Next slide. So we've screened almost the entire library of FDA-approved compounds. We just have a relatively small number left, and we're going to continue to screen compounds until we run out of money. So
wow.
once again,
that's really impressive. All the FDA library of approved compounds, because that helps us. You know, if you find something that resonates as you've done, it answers so many questions.
Yeah, it's actually a relatively small number of compounds. Roughly 6,000 drugs are approved by the FDA for clinical use, and many of these are kind of copies of one another. So we've tried to screen most of them, or at least ones that represent other cell classes. So it's just not economically feasible to screen large number of compounds. Next slide. I wanted to point out from data that the average cost to move a new molecule forward is $1.5 billion for a pharmaceutical company. So if you're screening unknown compounds, that's how much it costs to move forward. That's why we and others have taken the approach of trying to work with compounds that are already approved by the FDA, or that we have a good idea of how they're working molecularly, because that cost would be driven down considerably. Next slide. So have you tested whether the topo inhibitors could treat model mice? If not, why not? We haven't, because our current compounds aren't getting enough unsilencing throughout the brain, so we do not think they would be successful at this point. So our time would not be well spent pursuing whether we're getting behavioral rescue yet. It'd be likely to fail. I don't want to do an experiment that's likely to fail. Instead, our time is better spent finding topoisomerase inhibitors with optimal brain penetration and minimal off-target effects. So that's where we're putting our efforts, and that's why we're doing it. Next slide. So how likely is it that topo inhibitors will be taken into a clinic, and what's the time frame? I hate this question. I'll tell you why. The answer is somewhere between a year and never, and that's a pretty big gap, right? I know it's a very unsatisfying answer for a parent to hear, but that's the truth, at least in my opinion, that it could be as soon as a year, it could be never, and that is why I don't like answering that question. It's an impossible one to gauge, at least with the level of progress we have now in the research. I can tell you that as this schematic indicates, the path to taking something from an idea to new medicine is long and winding and tortuous. We try to shorten that path by working with things like FDA-approved drugs when we can. Okay? This is just a schematic I adopted from just took off Google Images, came from a pharma site, where this is showing that from a screen of 10,000 compounds, it might take 15 years to get one compound approved by the FDA. Okay? So just to also give you an idea of what a time frame could look like on average. We're obviously trying our best to shorten this time frame because we know that you want that. And by working with FDA-approved drugs, that's one of the ways we can try to do that. Okay? Next slide. So I also want to point out that working in academics, we would like to engage pharma. Okay? So if we get the might of large pharma behind this research, that will help us. They're less motivated to do it because, unfortunately, for you guys, the incidence of Angelman syndrome is so low that it makes it not a good moneymaker for them. So they've shunned it. But this is starting to change because there's such a pressure for pharma to have breakthroughs in neuroscience that they're incentivized to tackle problems that they think are tractable. Fortunately, Angelman syndrome is a tractable problem because the gene is known and the intact paternal allele of UB3A is there. So next slide. So I recently formed a partnership with Genzyme, which I'm really excited about. They're a very well-respected pharma company. They're a Sanofi company. And I can't, unfortunately, give you any of the details about that collaboration because it's protected. Property is protected intellectually. But I can tell you that I'm excited about this partnership because we can do more working with pharma than trying to fight against pharma. Next slide. So I recently got a grant to study the optimal treatment windows of when to treat Angelman syndrome and why and what behaviors could be improved. And people are wondering, well, look, this is useless because we do not yet have a treatment in humans. So why are you looking for optimal treatment windows? Now, I can tell you, I think this is actually extremely significant research. But to understand why it's important, you need to know why most clinical trials fail. You can Google this. Just Google why clinical trials fail. There's great literature on it. This is one website. Next slide. Many trials fail because of poor trial design. Next slide. Because the clinical trial is not properly designed or run, or the wrong clinical endpoints are selected. So to take an extreme example, a compound is being tested for Angelman syndrome. They're testing it in Angelman syndrome because they think it's going to improve seizure activity in an individual with Angelman syndrome. So that's what they put forth to the FDA. I'm going to test drug X to approve seizures in an individual with Angelman syndrome. Well, you put drug X into an individual with Angelman syndrome. It doesn't change their seizures at all, but they gain the ability to have speech. Guess what? That clinical trial failed. Okay? Because they put forth the wrong clinical endpoint. So the whole trial would have to be rerun with speech and communication as an endpoint, not seizures. So you have to go through that, all that expense of recruiting individuals and the time it takes. So if through a mouse model, we can understand when you should intervene to treat Angelman syndrome, and when you do intervene, what types of endpoints we can expect to fix, whether it's seizures, whether it's communication, whether it's sociability, whether it's motor issues, then we can design our human clinical trials more rationally. So even though this is basic research, it is going to help guide every single clinical trial in Angelman syndrome. So that's why I think it's important. So our study is going to determine when the treatment windows are and what behaviors can be fixed. Next slide, next slide. And this will guide rational endpoints for clinical trials. Now, there's also some concern that my lab is trying to do Rett syndrome research, which I think if there are any Rett families in the audience, they'll hopefully appreciate that my lab is doing Rett research. There's a concern that's taking away from the Angelman syndrome research. This is simply non-factual. In fact, my lab has been studying glutamate receptors in the brain. I'm phasing out that line of research in my lab, and I'm replacing that with Rett research. There's also other reasons to believe that it's important. Rett syndrome from this slide has many, many similarities to Angelman syndrome. Okay? Angelman syndrome and Rett are phenotypically have many, many similarities. So we have every reason to believe that the brain pathways that are affected and the molecular pathways that are affected are likely to be similar between Angelman syndrome and Rett syndrome. So by studying one, we gain insights into the other. So by studying both, it's likely to be mutually beneficial. Next slide. This isn't just my naive view. Here are a few recent studies showing links between UB3A and MECP2, which is a Rett gene. So you can see just from this small sample of literature, there's a very strong link between the two. So by studying one, you have ability to gain insights into the other. Next slide. I think this is my last.
I'm just actually so delighted because I've learned so much and it's so very well thought out.
So does your lab collaborate with other research to advance our understanding of Angelman syndrome? I think there's a concern amongst the parents that all the scientists are working in silos and none of them are communicating with one another to advance research. This couldn't be further from the truth. This is a list not just of individuals but of entire labs. That this first list is of people I'm collaborating with at UNC. So there's represented there is expertise in drug screening, molecular and genetic approaches, pediatric neurology, electron microscopy, two-photon microscopy, medicinal chemists, pharma, you name it. We're collaborating with these people at UNC. So once again, this is not my lab. This represents about 10 other labs just at the University of North Carolina that I've engaged in with my Angelman syndrome research. And just from looking at my publications, you can see that I've already published with many of them. Next slide. I'm also collaborating with many individuals outside of the University of North Carolina. So EPL Gersman and I are working to find out the critical treatment windows for Angelman syndrome. I'm working with Rich Mooney at Duke University to do imaging through the brains of the Angelman syndrome model mice to see what's going on in live imaging experiments. Youngwei Zhang at Duke developed the first Angelman syndrome model mouse, and we're collaborating. Stormy Chamberlain, who has the stem cells from the Angelman syndrome patients, we're also collaborating. There are others here who I'm not going to list, but we're collaborating extensively. We're not working in silos. And also I mentioned my collaboration with pharma. So we're really not working in silos. Now, not every scientist collaborates with every other scientist, and this is actually a good thing because that gives an opportunity to, one, for us to replicate one another, which is really, really important, and two, that people might come up with a different idea. So if too many people, if everyone's working together, we might kind of funnel into the same research path. So it's good to have both collaborations and some parallel efforts going on. I wanted to end on, I know it says last, end on a note of optimism. We've shown you that UB3A can be unsilenced in vivo. The unsilencing is long-lasting. A big caveat is this is just in these spinal cord neurons. We don't know whether we can get unlasting unsilencing within the brain itself. It can work in patient-derived neurons, and these really represent clinical candidates. So it also suggests that other compounds or approaches may be able to unsilence paternal UB3A. So even if this ends up being nothing more than a proof of principle, it's inspired us and others to pursue this as a treatment avenue for Angelman syndrome. So even though I'm sorry to say that we're not ready with the treatment that's ready for individuals with Angelman syndrome now, I do think that increased basic science discoveries, the increased interest and research into Angelman syndrome, the pipeline of putative therapeutics, and the engagement of pharma all hold incredible promise for the eventual treatment of individuals with Angelman syndrome. And I, of course, would be remiss if I didn't acknowledge, next slide, all the members of my laboratory here. Approximately, well, most of them are working on Angelman syndrome, and the remainder are working on Rett syndrome. I also have funding from the Angelman Syndrome Foundation, the Simons Foundation, National Institute of Mental Health, and the Rett Syndrome Research Trust. So I thank you for your time and attention. I apologize for going over.